speaking today with Omar Ahmed. He's an associate professor of psychology, neuroscience, and biomedical engineering at University of Michigan. I believe we first crossed paths at one of John Wasliff's excellent annual psychedelic therapeutics and drug development conferences earlier in the year, if I'm not mistaken.
That's right. And
Yeah.
and then again at the Gordon Conference for Psychedelics.
Yes, yeah, the GLC meeting recently, yeah, which both excellent meetings. And yeah, thanks so much for taking the time to discuss some of your science.
My pleasure.
Yeah. So I think you're obviously doing some pretty interesting, really fundamental sort of neuroscience research on the mechanisms of action of psychedelics. It spans a number of different sort of disciplines and interests. But I was wondering if you could give us a sort of brief background on how you ended up studying psychedelics at all and what your sort of motivations were for the science.
Yeah, when I started my lab, we had zero intentions of studying psychedelics. Let's just say that. So that was about 9 years ago. Um, we study memory circuits, we study Alzheimer's circuits, we study Parkinson's circuits, epilepsy circuits, depression circuits, but not psychedelics until about 6 years ago, when Tyler Eakins, a really talented postdoc, joined my lab and said, I really want to understand how psychedelics work at the neural circuits level. And I said, that's great, but as long as you can apply it to the brain regions I also care about. And that really got us going. And now psychedelics has pretty much taken over most of our lab.
Yeah, excellent. I mean, yeah, pretty fascinating. I think Alex Kwan has a pretty similar sort of origin story in terms of it being
Mhm.
a secondary interest to regions of the brain he was interested in. But some incredible work has come out of this already from your lab. I don't know, I know there's other manuscripts in the works. One of the things that I sort of note is that, and I'm not sure if it's a streetlight effect kind of problem, but many people are very interested in what psychedelics do in terms of their effects on sort of postsynaptic receptors, 5-HT2A and other receptors in the sort of frontal cortex of mice, humans in some cases. But you've done some pretty interesting work that shows effects on presynaptically located 5-HT2A receptors. Is that right? I was wondering if you could tell us a little bit about that work. So I thought that was quite interesting.
Right. So, as you know better than anyone, the story goes that psychedelics are therapeutically beneficial because they act on postsynaptic serotonin 2A receptors and cause more synapses to be formed onto those neurons. But that theory requires that postsynaptic neurons have 2A receptors, serotonin 2A receptors. Um, and it just so happened that one of the brain regions my lab really cares about, and you can actually see it in my background, it's called the retrosplenial cortex. And we were curious, how do psychedelics affect this brain region? But when we started looking carefully, we noticed that many of the neurons in this brain region completely lack serotonin 2A receptors.
Hmm.
In fact, we identified a little neuron that you can see behind me. The grad student who discovered it kind of called it the little neuron that could, because it loves to keep firing a lot and seems to be really important for memory and orientation and fear conditioning. But it has almost no serotonin 2A receptors at all. So we still went ahead. We gave these mice a single dose of psychedelics, and a few days later, we got a therapeutic boost in synaptic activity and number of synapses onto these neurons, even though they have no serotonin 2A
Hmm.
receptors. So that got us just scratching our heads a little bit, like, how could this be? The theory says that it needs to be acting only on neurons that have this receptor. And then, as you just mentioned, we realized that it doesn't necessarily have to be the postsynaptic side. It could be neurons that are projecting to these neurons, the presynaptic neurons that could have serotonin 2A receptors. So we built a new mouse line where we could get rid of these serotonin 2A receptors in those presynaptic neurons, got rid of them, and then when we did get rid of them, the therapeutic synaptic boost went away. So the bottom line being that serotonin 2A receptors are clearly still important for therapeutic effects of psychedelic drugs. But where they need to be located for them to have this therapeutic boost is more complicated than initially thought.
Yeah,
It can be on the presynaptic side as well.
yeah. I think the focus has really been on these sort of key pyramidal tract neurons in certain layers and the sort of postsynaptic 2A receptors there. But this was the first time I was made aware of the interesting role of sort of presynaptic 2A receptors in other sort of neuronal circuits. So
Yeah.
are you continuing to sort of dive into the mechanisms there and study, obviously psychedelics hit a number of different targets. Are you continuing to dig into the role of potentially other 5-HT receptors in other parts of the brain?
Yeah, we are. And even in this part of the brain, there's still many questions to ask. But I think the main thing we've realized from this paper and subsequent work is that psychedelics can alter far more neurons than we previously expected.
Mhm.
This is a brain region that essentially should not have been changed with psychedelic drugs, given the fact that it doesn't have 2A receptors, but it's profoundly impacted. And many neurons don't have 2A receptors. In fact, only a quarter of all neurons in the brain, of the mouse brain at least, do.
Right.
But the majority of all neurons in the mouse brain receive inputs from other neurons that do have 2A receptors. So now instead of psychedelics targeting a quarter of the neuron population in the brain, we're saying potentially that could be up to like 75 or 90%,
Right.
depending on which neuron has the post or the presynaptic 2A receptors. So we're continuing to figure out if the same rules apply in other brain regions, as you said, but we're also examining the role of 2C receptors,
Mm.
because the little neuron that could doesn't have any 2A receptors, but it's loaded with serotonin 2C receptors. In fact, some of the highest concentrations of serotonin 2C that you will ever find in the mouse brain.
Right.
So we've built a new mouse line for that, and that work is in progress. And we're now examining whether 2C could be just as important as the presynaptic 2A receptors
Right.
to lead to
But
these psychedelic therapeutic boosts.
yeah, that's awesome. But when you say a new mouse line that you're
Mhm.
generating, is this sort of a conditional knockout of the 2C receptors as well?
Precisely,
Yeah,
precisely. So
super
we just built
exciting.
that new mouse line, and 2C is a sex-linked gene. So that took a little bit longer, but that mouse line is working well now, and hope to share that with the world in the coming few months.
Yeah, very cool. Super cool stuff, Omar. I'm a huge fan of these sort of conditional knockout lines, and I know they take quite some time to generate. Are you planning on taking this sort of as far as you can across a number of other 5-HT receptors, or are you pretty satisfied with sort of 2A and 2C for now?
I think we want to go further,
Right.
so I mean, together with the whole field, it would be great if we and others could team up to come up with a cohort of genetic lines that allow anyone to manipulate any one of these, really half a dozen to dozen serotonin receptors, so that we can systematically figure out the fundamental rules of what does and does not lead to therapeutic boosts from psychedelics, because so many of these receptors are just really poorly understood, including 2C.
Right. Yeah.
I mean, 2C is physiologically like it's a mystery. And I'm just stunned kind of looking at the literature that I can't find the answers that we're looking for. So we just have to do those experiments sometimes ourselves. Just physiologically, what does 2C do? Where does it act? Is it more important presynaptically or postsynaptically? Those really basic fundamental questions are hard to answer, and we need to answer them so that we can understand how psychedelics fundamentally work.
Yeah. Very cool stuff. I know that one of the sort of interesting things I saw in some of your recent papers was also a bit of a focus on behavior, not just this really fundamental
Hmm.
mechanistic neurocircuitry view of what psychedelics are doing,
Hmm.
but also their effects on behavior. And I noted in a recent publication, you were looking at the effects of a compound that might not be known to too many people called 25-CN-NBOH.
Hmm.
So for those who are not aware, this is essentially the free hydroxyl N-benzylated phenethylamine of a sort of analogous compound that would be 25CN N-bomb. So this is a compound that's contains a methoxy group in place of the hydroxy. It's been used as sort of a radio tracer as well in carbon-11 labeling for PET. But also some of these N-bombs have been used recreationally and they're quite toxic, right? So Brian Roth and I have spoken about
Hmm.
why they might be apparently more toxic than the corresponding phenethylamine
Hmm.
analogs or other psychedelics. And there's a few different hypotheses around this that I don't want to get into here too much, but the 25CN MBOH is interesting because it's quite a lot more selective for 5-HT2A,
Exactly.
right, than other compounds. So I really like this idea that you can start using a more selective tool compound, and we've discussed using this ourselves. Do you know, I went looking online because there's a little bit of data on Reddit and other places suggesting that these sort of analog compounds 25E and 25C
Hmm.
NBOH are actually psychedelic in people,
Hmm.
hallucinogenic in some form, but I couldn't find any information for 25CN NBOH. So do you have any idea if it has been confirmed to be a psychedelic in people or not?
Yeah, I think the postdoc in the lab, Tyler Eakins, went looking for that information as
Mm,
well, and did find occasional anecdotal references
right,
where apparently it's been consumed and is thought to be psychedelic, but it's,
right.
you know, take it with a grain of salt.
Yeah, that's my view of the entire Reddit body of knowledge is that it's
Exactly.
anecdotal, but it provides some evidence, right? And there's not a lot of posts on any of these compounds.
Exactly.
But this suggests that you can have, given the analogous sort of 25C and 25E NBOH
E.
compounds, which are also reported to be hallucinogenic, it seems clear that you can definitely have highly selective 5-HT2A agonists that are anecdotally psychoactive in people. But you've used this as a research tool in looking at the ability of psychedelics, and I guess in this case, specifically 5-HT2A agonists,
Precisely.
to modulate sort of cognitive flexibility in rodents. So I was wondering if you could tell us a little bit about that.
Yeah, and we used it for precisely the reason you laid out, which is that it selectively has a much higher binding affinity to 2A receptors than it does to, for example, 2C. And we love that ratio so that we can try to isolate 2A versus 2C effects here in particular.
And just for context, at this point, I would say many labs have now shown quite a robust effect, I would say, on various cognitive flexibility type behavioral paradigms in rodents using things like psilocybin, right? So other psychedelics. But what I liked about your study is you've used this highly selective 2A agonist.
Right. So we used the highly selective 2A agonist. We measured cognitive flexibility. And I think one of the things we did slightly differently was we had a slightly harder cognitive flexibility task, where it's not that hard for even rats would find it easier, but mice can take a few weeks to learn it. And then we had them essentially reverse the rules and learn those. But because that takes a while.
Who are not familiar with these sort of paradigms, what's typically involved in either the mouse or the rat case, just so people understand?
Yeah, so essentially mice need to learn a very simple task, which is press a left lever and then the right lever, and you'll get a reward. And then suddenly, 3 weeks later, we will reverse the rules for them. So now they'll have to press the right lever and then the left lever to get a reward. Sounds easy enough to quickly switch over, but it can take rodents quite some time to kind of get over the frustration and learn that the rules have completely changed. And that's why it's called a cognitive flexibility task, because you have to think about this more flexibly, that it's a reversal learning task as well, as sometimes what it's called. The rules themselves can change. You're still doing the same kind of work, but under different conditions and different rules. The important thing here was that the reversal wasn't happening till 3 or more weeks after the psychedelic single dose of 25 CN and BOH had been given. So it's a single dose, and 3 or more weeks afterward, up to 3 weeks afterwards, we're looking at reversal learning. And even up to 3 weeks afterwards, we see improvements in reversal learning and cognitive flexibility in these mice after a single dose of psychedelics. So I think that makes a lot of sense because, I mean, when you look at the therapeutic potential of other psychedelics in people in clinical trials with people with major depression, those effects are lasting many months. When you look at Alex Kwan and other people's works looking at how long the synaptic boost onto neurons lasts, that can last many weeks in mice as well. So what this is kind of showing is that those synaptic and cellular correlates of psychedelics that are more and more recognized and well established are also accompanied by these behavioral effects that can last multiple weeks after a single dose, which is important to just confirm.
Yeah, I think it's incredible work, and I'm a huge fan of these sort of cognitive tasks in rodents because these aren't just experiments in mice and rats, right? There are correlates that we can actually clinically test for cognitive flexibility in people, and obviously rigidity of thought and some of these other sort of cognitive inflexibility type symptoms are associated with different psychiatric illnesses, right? So there's a real sort of therapeutic translational potential here.
That's right. Yeah, absolutely.
Yeah, super cool work. You use 25C and MBOH just because you wanted to use a more selective agent, but what other psychedelics are you typically using in your lab?
We continue to stick to more mechanistic questions. I think the whole field is doing amazing work with psilocybin and DMT and many others. We like 25CN NBOH because it is such a selective 2A agonist. And when we're targeting 2C, we prefer DUI over other psychedelics because that is not perfectly selective for 2C only, but as far as other psychedelics go, it's probably the best one out there for that question. So we're happy to ask these questions all over again once we figure out the mechanisms with these two particular drugs using other psychedelics. But our goal right now is to systematically, mechanistically, and behaviorally understand how these two drugs end up impacting neural circuits and behavior.
Yeah, incredible. Yeah, I like this really sort of reductive approach that gets down to the mechanism and avoids some of these confounders with the sort of challenging polypharmacology of psychedelics.
Right. Polypharmacology is a challenge, and the more we can isolate certain receptors, the better.
Yeah, totally agree with you on that one. I know you have a number of manuscripts probably under review right now in some nice high impact journals, but is there anything else you want to discuss that's in the works?
Yeah, so mechanistically, we've got some work under review that shows that psychedelics don't boost neurons in terms of their activity patterns, but they actually suppress them.
Hmm.
So we have a preprint out on that, which anyone can go and find and read. And we're finding that psychedelics are actually acting on ion channels that are in practically every neuron, independent
Oh,
of the 5-HT2A receptors. So again, the preprint is
interesting.
out there, so people can go find it.
Which channels are these?
Yeah, it's a potassium channel. It's a potassium channel that actually opens up and makes the neuron way less excitable. And it works at the lowest possible doses that we can give in these conditions.
Oh,
And
amazing.
yeah, so it's the effect that actually is the dominant effect in our hands on individual neurons. And we think that it can be hard to find in vivo because you get all of these other competing effects on other types of neurons, inhibitory neurons. There can be disinhibition, there can be many other things happening. But
Right.
when you look carefully at individual neurons, you can see that this is the dominant effect, suppression of excitability of neurons.
Fascinating.
And the analogy I like to use is that people have always said that psychedelics excite neurons, and that is partially true and definitely true. But if you think of a neuron, perhaps think of it as a pole vaulter trying to jump over a bar. What psychedelics do is they move the floor closer to the bar, so you can jump over the bar more easily, and that's been appreciated. But what they also seem to be doing is they literally take away the pole. And that's this opening of potassium channels.
Hmm.
When you take away the pole, that pole vaulter is not getting over the bar. And the analogy is that the neuron is not going to be able to fire and activate.
Right.
So we have these multiple competing things that happen simultaneously. And I think it's really important to take a mechanistic and computational viewpoint and understand them all together. So we ended up building these fairly detailed AI-based machine learning models to kind of integrate all of these possibilities and see what the net result is. And the net result is still suppression.
Fascinating. Yeah. Yeah. Absolutely incredible work, Omar. And I really enjoyed your presentations at the last few conferences, and I'll look forward to seeing you at the next conference.
It's always a pleasure to talk to you, Sam. So thanks again for this opportunity.
No, thank you so much for your time. Great speaking with you, Omar.